This video is about cell division, and what we're going to be looking at is what we call mitosis, which is part of really a larger process called the cell cycle. So in this video, we're going to look at how we make new body cells. And so, of course, we all began as a fertilized egg or a zygote, and that egg split, or that cell split into two cells, and then these two split into four. And then that division continued until, we ended up with what we look like today. So cell division for us is going to be used in multicelled organisms for growth and development or repair and replacement. So, think of all the times that you've injured yourself or cut yourself or something like that. So cell division is going to fix those wounds for you. At the end of this presentation, I'm going to talk about asexual reproduction and how cell division is used by single cell things like this amoeba to reproduce. But we're going to focus on growth and development and repair and replacement for just a moment. When we look at a cell that needs to divide, remember, we want the new cell to be just like the old cell. So that's why we copy everything in the old cell first before we divide it in half. And it's a big deal because there's a lot of stuff inside the cell, and we need to make sure that exactly half goes, into one cell and the other half of the stuff goes into the other cell. So we need to make sure everything is done the way it's supposed to. Of course, the most important thing we're going to copy is the DNA, which is located inside the nucleus of the cell. The DNA in this picture is drawn, it really just looks like a bunch of squiggle lines inside the nucleus. And what that's representing is the state of DNA in a cell that is not dividing. So when our cells are just doing their regular thing, functioning normally, meaning they're not dividing, the DNA is unwound, and we don't even have chromosomes at this point. So it's like all the chromosomes have been, totally pulled apart and unwound, into this tangly, stringy mess. But this is actually good because this is how we need our DNA to be for us to make proteins, which will be what we talk about in our next chapter. But when it's time for the cell to divide, the DNA is going to go from this state, which is just all unwound and all tangled up, into chromosomes. So you can see the difference in what's in the nucleus in this picture and this picture because the DNA has wound up. How does it do that? It uses these little pink dots here. They're called histones. They're proteins. They're part of the chromosome itself, and they control if the DNA looks like this or if it looks like this. Now remember, when the DNA looks like this, we call it chromatin with an N on the end. So N like night or something like that. So chromatin. When chromosomes are formed, especially if we're thinking about our chromosomes, which let's do that for just a moment, we are going to have 46 chromosomes in every single cell in our body. So all of our normal body cells. So if you think about one skin cell, it has 46 chromosomes. Those chromosomes are paired up. So remember that this chromosome, may have come from your mother, and this one may have come from your father. And they have the same information on them. And this is called a homologous pair. So, the prefix homo, H-O-M-O, in science means same. So there's the same genes on this one as there is on this one, okay? And we have a homologous set of chromosomes, or a diploid set, probably the more accurate thing to say, in all of our regular cells. So these are homologous pairs, and this is a diploid set of chromosomes. Diploid means that you have two of each. So remember, di is a prefix, means two of each. Of course, this is the karyotype of female because you have two X's down here. Okay, same thing down here. So this is the chromosomes of a male. So remember, each one of these came from each parent. This is a homologous pair. and this is, as a whole, a diploid set of chromosomes. And then we're going to have diploid numbers in all body cells. Okay, so just to kind of back up and talk about DNA for a minute, DNA is shaped like a double helix. It's made of a sugar and phosphate backbone, which is what you see here. Sugars are going to have a shape, and so these are 5-carbon sugars, so they're shaped like pentagons. Of course, over here's the phosphate. So if we think about DNA as a twisted ladder, the sides of the ladder, the very sides over here, that would be made of sugars and phosphates. The nitrogen bases are going to be in the middle. And of course, this is going to make up the code that we look at when we're talking about genes. These bases are paired up, and so remember, A always pairs with T, C always pairs with G. Because remember, apples go on trees, cars are parked in garages. And these little pink dots or red dots that you see here, these are hydrogen bonds. So you need to know that, okay? Now, if we look at the sugar and the phosphate and the nitrogen base together, that's a nucleotide, okay? Watson and Crick, remember, discover the structure of DNA. They got the Nobel Prize for that. They got the Nobel Prize along with Maurice Wilkins. So remember, he and Rosalind Franklin were taking pictures of DNA. Remember, Rosalind Franklin died before the Nobel Prize was given out, so she didn't get it. But Watson and Crick are the two guys that are given credit for discovering the double helix structure. Okay. Now, when DNA is copied, this is going to occur at the very beginning of the cell cycle, and it needs to be copied correctly, and so that's a very big deal. And when we think about cells that are not normal, when we think about cancer cells in particular, that's one of the things that happened. The DNA was not copied correctly, and that may have been what caused that cancerous cell to form. So we'll come back to that, but just remember, the DNA is a big deal to copy the DNA correctly. Okay, we're not going to talk about that just yet. Okay, so when we look at cell cycle as a whole, we have 3 big phases. We have interphase, then we have mitosis, and then we have cytokinesis. Okay, and so the main thing that happens during interphase is that we're going to copy the DNA, which is what we were just looking at there. That is going to occur during S phase. So interphase has 3 different phases by itself, which is G1 and S and G2. Okay, so what we see again here, a single chromosome is what we have at the beginning of interphase. By the time we end interphase, we've got a double chromosome. This double chromosome is made up of 2 chromatids. So this is a chromatid with a D on the end, like D for dog. So this is a chromatid, and this is a chromatid. And it's joined together by a centromere right here. Okay, so this is what we had before S phase of interphase. This is what we have after, okay? And so that's why when we start cell cycle, we have a cell that has single chromosomes in it. And then after we start cell cycle and after we complete the first part, which is interphase, we have double chromosomes, okay? So during interphase, we have 3 stages. We have G1, and then we have S, and then we have G2. Remember, G stands for grow. So G1 we grow. S stands for synthesis, and that's when we make more DNA. So that's when we make these double chromosomes. So during S phase, we go from singles to doubles, okay? And then we complete the whole interphase with G2. That's when we make more organelles, okay? Notice we still have a nucleus. So we're through with interphase, which remember takes a really long time. It takes 90% of the cell cycle. Then we move on to mitosis. And so mitosis begins with prophase. During prophase, we still have a nucleus, and we have double chromosomes inside the nucleus. And in fact, this is when the chromosomes form. That's why it says wind up over here. So we go from stringy DNA to DNA that has wound up into a chromosome. Okay, so that's what prophase is like. Next phase is metaphase. So we have no nucleus during metaphase. We have our chromosomes, they're doubles, lining up in the middle of the cell, okay, in the equator of the cell. We have our centrioles over here on the poles of the cell. So think like light pole or something like that. So the ends of the cell, the poles of the cell, and then we've got the chromosomes in the middle. These green lines are spindle fibers. That's metaphase. Anaphase, we pull the chromosomes apart. They're now called chromosomes. So now they're single chromosomes. And so you see all this space in the middle because we're wanting half of everything to go to this side, which will be one cell. Another half to go here, so that's anaphase. And then we have telophase, where our nucleus comes back and we start to divide. Remember that in cytokinesis in an animal cell, you're going to have a cleavage furrow form. So this is in animal cells. You don't see this in plant cells. In plant cells, you have a cell plate, Plate, okay? After these divide, so this will be one cell, this will be the other cell, then we end up with daughter cells, and they're just like the parent cells, okay? So this is what it actually looks like in animal cells, and there's some in plant cells, okay? So again, if we look at cell cycle as a whole, we start with interphase. That's why you see this I up here. That's when we do G1, S, and G2. S is the important phase. S is when we make an extra copy of our DNA. So our single chromosomes turn into double chromosomes. Here's prophase. We wind up into visible chromosomes. You can see them here. So here we're starting or trying to go to metaphase. So this is the end of prophase. So our nucleus is going away. Our doubles are getting ready to line up. Then we pull them apart, and then we chop the cell in half. Okay? So that kind of wraps us up for mitosis. Now, if cell cycle goes terribly wrong, then someone may end up with cancer. And the reason that they get cancer is because the cells divide really out of control. And they divide when they're not supposed to divide. So cells have chemical signals that tell them when it's time to divide and when it's time to not divide. And different cells divide at different rates. So you can see some of the, how often these cells divide. Nerve cells, and this is mature nerve cells. So this will be like my nerve cells. They are permanently in G0 and they're not dividing. Your brain cells they're still dividing. Your brain is still developing and you're still making new connections. And so that's why your brain is very vulnerable, because nerve cells, once they die, they don't really come back. And so when we look at, the effects of drugs and alcohol, that has a much more significant effect on a nerve cell than another cell of the body, and especially nerve cells in people that are still developing like you are. So when we look at what's going on and how cancer occurs, it means that a checkpoint in the cell cycle, it was failed. And, think about the doors that we have on our school building. So, especially the door between the band hall and my classroom. If you're at Rosa, there's a little box on the wall, and if you don't have the key card, it's not going to open. So that's a checkpoint. So people without a key card cannot come into our building. So cell cycle has that too, and it's just a stop and a go. So it's like, okay, is everything okay? Can we go further, or do we need to stop and reevaluate and not do cell cycle? So you really don't have to know these, but there's a G1, a G2, and a spindle checkpoint. So these are just points in the cell cycle. They're stop and go. Is everything okay? Can we go forward? Is everything not okay? Are we going to stop? And so that's what that means. If if this checkpoint says go, then the cell is going to divide. And then, of course, if you fail it, then it's supposed to stop. G0 is when you have when your cell is just not doing cell division. So it's just most of our cells are going to be in G0. Of course, they don't hit G1 until it's time for them to divide. So we said that cell division is triggered by chemical signals. They're just go-ahead signals that tell it it's time to start the cell cycle. Cells only divide when they need to. And so if you look at these pictures over here to the side, this is what our cells normally do. So if you have normal mammalian cells, just regular cells, you put them in a petri dish, they're going to divide until they fill it up. Okay? If you take some out, they're going to divide to fix it. So it's just like we're just filling in the gaps, and that's it. Okay? So we're just going to make what we need. If cells are touching each other, then they say that it's time for the division to stop. In cancer, that doesn't happen, and that's because you have messed up genes. Sometimes the genes are just normal genes and they turn into cancer genes, and sometimes you just have cancer. You're just born with cancer genes. So when we look at those, again, they're controlling cell cycle. P53 is a big cancer gene that is very involved in many different types of cancer. So cancer means that all these things are going to happen. So you have unlimited growth. So look how now the cells are filling up the dish where they just made one layer earlier. The checkpoints are ignored. Apoptosis doesn't occur when it's supposed to. When we make a bad cell, normally those cells will dissolve themselves. It just doesn't happen in cancer cells. Then you get lots of blood vessel growth, which means that you're taking more nutrients to the cancer cells. And so just all kinds of things are going out of control. Several things that cause cancer, which you know these: sunlight, which is UV radiation, cigarette smoke, pollution, AIDS, age genetics, all kinds of stuff there. Down here, they're just showing you breast cancer because, breast cancer is such a common form of cancer. If this is the tumor, of course, it can grow. If the cells from this tumor end up in the lymphatic system, it can travel to other places, and that's what metastasis is. So if cancer metastasizes, that means that it travels somewhere else, and it spreads, which you've probably heard people talking about cancer spreading, and that's what that means. Benign means that you have a tumor. It's abnormal. It's kind of like freckles or moles, things that we're not worried about. Malignant means they're bad. And so malignant tumors and the cells from malignant tumors, they can go and travel other places. Of course, if someone gets cancer, there is